Hi friends, and welcome Harish Chandra Anandram sir and our viewers to Joel John J YouTube channel. And today sir will give an insight about artificial intelligence and biomark prediction. And also sir's ebook is being distributed to these video viewers for free. Sir's WhatsApp number will be available in the description. That's you have to scroll down the video and click the more tab and text to share sir's WhatsApp number. Only text, no phone calls, and no marketing and forwards and spams, please. Sir will be sending you the free ebook. And also, I have published a book, but that's a paid one that is regarding social media and content writing. So you can scroll down to look at the other links, and also at the end screen, the entire playlist of Sir's videos will be there. Click on that and watch that as well. Okay, friends, over to you, sir.
Well, thanks for the detailed introduction. And as per the today's topic with respect to the artificial intelligence and the biomarker prediction, first we need to understand the computational pathology biomarkers. Most people might have studied pathology in medicine. And computationally, what and all we require here is here the diagnostic system contain vast amount of data. So we predict the treatment response and the outcome, and then the patient stratification, that is the level of recovery and relative aspects, the selection of treatment, and the cancer epidemiology. Epidemiology involves the study of a particular disease with various regions. The impact of the same disease in a different region is different. So the diagnostic system is tested basically with the tumor lymphocyte infiltration level, the cellular necrotics aspects with respect to the lymphoblast and the spatial distribution and the morphology of glands and ducts and the orientation of collagen. So these are the aspects of computational pathology based biomarkers. So with respect to pathogenesis, the people who might have used flow cytometry for understanding the programmed cell death called apoptosis would have found that the tissue related aspects levels the labeling of biomarkers, and there is a clustering aspect which is happening beyond the cells. You see there is a cluster of cells which defines the factor, and those underneath aspects are being studied as the TALs, which is also known as the tumor spatial based lymphocytes and the infiltration models associated with it. So, what and all studies we require here is the spatial TAL density to the prognostic value, because prognostic is the basis for the diagnosis, and then it can be seen in multiple cancers. And the programmed cell death process, which is not happening in cancer, is the key here. And we do have a standard TNM classification for the TALs. TNM is the classifying mechanism for the number of monocytes and the related tumor acrocytes model. So the potential clinical applications of TALS is already being published in 2014 itself by the Cancer Research Consortium, and a lot of artificial intelligence is also involved here. Why artificial intelligence? Because the data is not in 10,000s or hundreds; it's more than 1 lakh. Now the cancer uh, atlas contain around 5 lakh data of various patients in various levels. So we incorporate the TALs into a diagnostic criteria, and then we go for the predictive prognosis for survival. And the same combination is being taken for the immunotherapy. Nowadays, people treat with our own cells also. It's called as the immunotherapy. The potent cells called as the stem cells are being in research in cancer also. But the success rate with respect to cancer is relatively less. Autoimmune based stem cell therapy. Tumor infiltration lymphocytes, that is the TALs, were also being published, and deep learning based computational method is taken to analyze the biological strains. And they have tested it with the well-known TCGA dataset, which is called as the global studied dataset reference for cancer. And then you can see 13 subtypes of cancer were classified properly. Though there are more than 600 to 700 subtypes, the initial level of 13th cancer subtype classification is a good number. And then the detected TALs correlate with the pathologist who study on eye as well as the molecular estimate, because cancer might reflect the other organs also in future. So it's better to consult with the well-known experimental pathologist from the computerized data you get. And then we extend our research with respect to 20 subtypes of cancer. So now the Cancer Genome-Based AI Consortium is open to everyone, and whomsoever does a basic experiment or a simulation study can give the part of the outcome, and it will be extended to the further studies. So the term pathomics have come up. Before it was only genomics and proteomics. Now pathomics gives the tissue analytics for the precision medicine based on the single nucleotide polymorphism present in the genome. And the high resolution based detection is taking place here because you have a lot of advanced AI based minute cameras in the microscope as well as the confocal level microscopy and relative aspects to support the scoring of the tumor infiltrated lymphocytes and the composition analysis for detecting the tumors. As you can see very clearly here, from the normal image to this image, you can detect the red, blue, and green ones very clear. All these are possible due to AI. So the collaboration is not only with respect to NIH and WHO, it is also being extended to ECOG, a clinical trial consortium for extending the relative AI research. And you can see the collaboration of AI all over the world, not only in a specific place. So there are certain quantitative measures given by two people. According to the paper written by Arvind Rao, it is the agglomerative clustering and the relative cluster size and the distance between the relative cluster and the cells gives the index. And he created a package for the data analysis from the image data. He called it as C index and the determinant ratio. And there is another qualitative index given by Alex and Rajgupta. What they say is it is the band-like relative tumor lymphocytes, which is up to 30% more in one area and 5% in the relative area. And they have tested it with 1,083 samples. So it is only 1%, but that 1% of minute differentiation caused the major variation. You can see the cellular carcinoma diffusional pattern. You can see the normal image of complete clustering, and you can see the spatial distribution, the AI image in the last part. So the patterns of the TAL infiltration began with 10 initially, and then developed into the relative patterns. And they have done the analysis throughout the TCGA breast cancer based data set. And they have collaborated with the UNSC group also, a group known for studying blood-related disorders. And some unpublished results for the TCG analysis, they have initially incorporated in the websites. And then after experimental testing, they are going for the publication part. And then you can see the ratio of the index given. One index, they call it as Bell Hall Adjusted Index, that is the BRCA. And the statistical significant value is 0.07. As you can see very well clearly that till 0.05 is acceptable, but these values are very, very less than 0.05. So the level of significance is very high. And then a relative work is published recently in the American Journal in 2020 regarding the spatial distribution of tumor infiltration cancer data types with respect to AI. And then the risky feature associated with the initial data before AI was that the correlation index of cases, no, it is associated with either 0 or 1. There are no additive features. But in this AI-based thing, you have 2 plus additive features to get the reliable results. And then it is being developed as a community also, the tumor infiltration lymphocytes in breast cancer. And this community is also holding rights for cancer-related publication and sharing it with the WHO. And they are even validating the FDA-based collaborations because for the drug approvals, you require FDA and the relative aspects. So these tumor infiltration, lymphocyte detection and quantification by AI method is not the future. It's already happening, and it will be extended to every place in the future. And there are a lot of relative tools also based on the technology to quantify further. And they are also validating the AI methods. It's not just like AI, you predict something, you publish. You are validating with the clinical people also, and then going ahead with the relative pilot study. So they are collaborating with ICTR programs also. That is International Tumor Consortium Research, the National Collaboration for Immunology. So they are collaborating with radiologists. You can see the larger size of analysis they are doing in radiology. So you can see the level of work and the impact it has created. Around 71 collaborations across the country and relative data sharing in all the country. And the proof of concept is also successfully validated, and it is being deployed and published as a preprint. Soon, the work will be published as a main journal paper with complete validations. So you can see the quantitative results in various sites, and the data divided in training as well as validation. You can see a good correlation between the training and the validation set. And you can see all this data in the preprint. Soon it will be available for publication. And thanks to the research consortium for taking this effort. And thanks to sir for giving me an opportunity to present this video.
Thanks a lot, sir. And we have to thank you for giving a lot of insights on new fields and updates. And people who want sir's ebook, please contact him on his WhatsApp number by texting. And also, we have few more details like the podcast which we have done for the all the videos of sir's program, and that will be given in the more tab as well. If you want to listen only to the audio when you are traveling or when you are cooking or at gym or sometime, you can use that as well. And okay, sir, thank you very much for listening to the video. People, press the thumbs up and subscribe, enable all notification. And most probably now, as the end of the video, you'll be getting the playlist on the end screen. On that, if you click on sir's video, you'll be able to sir's playlist. You will be able to see all the videos which are given by sir, and also those details are also on the description in the more tab. Okay, sir, thank you very much for coming, and bye to you and our viewers, sir. Bye.
Thanks a lot, sir. Thank you. Bye-bye.